Hello and welcome to this session, Accelerating the Search for Drugs Against COVID-19. My name is Silvano Coletti. I'm the Chief Executive of Chelonia Applied Science from Basel in Switzerland and the Project Innovation Manager from Escalate for COV. I'm so glad that you can join us today. Just a few months ago, in this challenging time with pandemic, we Chelonia Applied Science were to introduce some important new projects, as I mentioned, Escalate for COV, that is a private-public consortium supported by the European Union. Chelonia is one of the 18 members of the consortium coordinated by Polypharmacology. And today we have got some more huge announcement for you. Probably you heard about that last July. Escalate for COV announced that to have found raloxifene as a drug molecule active against mildly infected patients. And 2 weeks ago, we were green lighted by IFA, that is the Italian regulatory body, to start a clinical trial on 450 patients in 2 Italian hospitals. I will tell you more later on today. All this in just 6 months, thanks to the exploitation of the best supercomputing resources available in Europe and artificial intelligence. I mean, this year, scientific research have become even more important in our daily lives, probably more than in the past. We understood that science is not an abstract entity. And we understood the importance of to be able to predict the mechanism of action of the nature to develop a vaccine, or as we are doing with Escalate for COV, to predict the mechanism of action of the drug in months instead of years. And we realized how magical is the equilibrium that grants us the life. Under the theme of One Word, Our Health, speakers here are placing a special focus on COVID-19, alongside, of course, other urgent healthcare challenges. But I want just to remember something that Professor Delord Darcy of Demon, Executive Chairman at World Innovation Summit for Health, said yesterday, say that it was time for a global pathogen shield to protect the world from novel diseases and a new global capability to rapidly develop new therapies and, of course, vaccine also. And I want to follow the remark from the executive chairman. And today I want to bring, of course, my passion and the Escalate for COV result to even more people globally, thanks to this great event, World Innovation Summit for Health. Yes, this is a journey. This is the virus commonly known as the coronavirus, which caused unprecedented damages throughout the world. It all started with a cluster of pneumonia cases, which led to the identification of the pathogen, and shortly afterwards, the viral genome was sequenced, and researchers around the globe could start their work to learn more about the virus. Unfortunately, the virus could not be contained and spread worldwide and was declared a pandemic in March, leading to increased effort to counter it. And here we are with the pandemic, fast forward to now. The virus has spread everywhere and keeps causing damages. So the key question is, why was or is it so damaging? And there are 3 points where we were focused on the research, and today we are focused on point 3, no drugs to fight it, how to find them quickly. Let me start this short animation. This is the workflow of the phase 1 of the project. The purposes with non-safe gases, food, ingredients, other natural substances, drugs from other institutions, and pharma companies. We worked on HPC screening in Silico using the state of the art, need to know about the chemicals and the viral proteins. And of course, after the screening, in this project we validated the results, going through trials, and hopefully with a new technique. Now we go for the workflow of phase 2, which is the clinical stage of billions of molecules, which means all commercial molecules plus anything chemical possibly and supply. And we have resulted in the workers leaders in clinical field. So doing that, we use the supercomputer center to do the calculation, and then we go through the usual drug development. From the proof of concept to the toxicology to again to the clinical trials and new stuff. But let me give you some facts today about the Escalate for CoV project. First of all, Escalate for CoV means exascale smart platform against the pathogens for coronavirus. And the objectives are identify molecules capable of targeting SARS-CoV-2, develop effective tools to counter future pandemics, and exploit European supercomputer capabilities. Some further information. We received a 3 million budget from the European Union H2020 program, an emergency call in response to COVID-19 in February 2020. And the duration of the project started in April this year, is 18 months up to September 2021. I mentioned 18 partners from 7 different countries. The 18 partners of the consortium have an impressive skills in the field of HPC, molecular modeling, biology, genomics, and big data, and they are Polypharmacology, that is, by the way, the coordinating member, Politecnico di Milano, Cineca, Università degli Studi di Milano, Catholic University of Leuven, the International Institute of Molecular and Cellular Biology in Warsaw, the Elettrosynchrotrone Trieste, the Fraunhofer Institute for Molecular Biology and Applied Ecology, l'Istituto Nazionale per le Malattie Infettive Lazzaro Spallanzani, the Barcelona Supercomputing Center, the supercomputers from Jülich Center, l'Università Federico II di Napoli, l'Università degli Studi di Cagliari, lo Swiss Institute of Bioinformatics, il KTH Royal Institute of Technology, l'Associazione Big Data, l'Istituto Nazionale di Fisica Nucleare in Italy, and last but not least, we as Kelonia Applied Science. And in addition to the consortium capability, we can count now on another important collaboration, such as pharma companies that provided the real samples by using our Dropbox, and other partners such as Eni, Nanome, D-Wave, SAS, and others that have put very important technologies at our disposal. If you are interested in the participation to our league, please visit this link, the escalateforcov.eu league, or ask me by email. And thanks to our partners, we have access to some of the fastest supercomputers in the world. Plus, thanks to the recent investment planned in Europe, new further systems are underway and will help us, Leonardo supercomputer, which will be hosted at Bologna Science Park, Italy, the Mare Nostrum 5 at Barcelona Supercomputer Center that the deep science of the project. So we aim to let the computer find drug-protein interactions, but let me first show you such an interaction with remdesivir as an example. The blob that you see on the left is a complex of 3 different SARS-CoV-2 proteins, which make up the RNA replication machinery. And you see the RNA template and the RNA primer, so the copied RNA. And you see the remdesivir that is in purple that mimics an RNA nucleotide building block and is interesting part of this picture. The host can produce viral proteins but cannot replicate RNA. Hence, the virus brings the RNA replication machinery with it. 3CL processes PP1AB and NSP7, NSP16 build the replication system. Both are prominent targets. Okay, so I think that you don't hear me. Okay, so I can go ahead. So both are prominent targets for drugs. For example, remdesivir binds to RP, as you can see in the picture, but unfortunately, no structures were known end of January. Now I have a video that helped me and support me and explain you how it works, the drug design.
Escalate for COV aims to exploit the Escalate platform, along with the most powerful computing resources currently based in Europe, to empower smart in silico drug design. Escalate for COV is a fully integrated network built to have a continuous run of in silico simulations followed by in vitro experimental validation, the speed of the identification of active compounds to be tested in humans as novel treatments for COVID-19. What you see right in front of you is a protein called protease, which is an extremely important one for the coronavirus. The peculiarity of this protein is that it generates new proteins and new peptides, which are of extreme importance of the virus. Thanks to the presence of these three-dimensional arrays, we can carry out detailed studies on an atomic level to discover which key points have to be bombarded to ensure that the activity of the virus protein is impeded. Thanks to the presence of these structures, we can identify the so-called binding site. In other words, an area of the protein which may be targeted using new drugs. In this case, as you can see, the molecular indication in blue is a ligand with the capability to interact with this part of the protein and impede its activity. Interact with this part of the protein and impede its activity. Thanks to these crystallographic models, we may acquire an in-depth knowledge of which areas we can strike with new drugs. By doing this, we are capable to go so deep that we can study atom by atom how the drug interacts with the protein, blocking its activity. In DOMP8, thanks to the ESCALATE platform, we can also study the protein from a dynamic viewpoint. Furthermore, taking advantage of the technology known as molecular dynamics, we can monitor over time how protein and ligand can interact with each other, thus taking advantage of the fourth dimension. As you can see in this case, our attention is focused on the protein and the ligand, that is to say, the entire complex, and we study it dynamically, not just statically, as can be done using the crystallographic structure. By observing the interaction between the ligand and the protein in dynamic terms, we can identify different states of the same protein that may be useful in the repositioning of new drugs. Another essential characteristic of our ESCALATE platform is that of offering a database containing billions of molecules that can be evaluated regarding inhibitory activities against virus proteins. Therefore, due to our capacity to use an extremely high number of new molecules on hundreds of structures of the same protein, we may carry out massive virtual screening and molecular recognition studies. In this way, we can compare a huge number of small molecules and proteins, obtaining very important information on new potential drugs. This will therefore lead us to identify which chemical entities will be able to recognize and interact with the new viral proteins, thus blocking their activities and vital functions for the virus. Hence, thanks to the enormous computing power of the ESCALATE platform, we will be able to effect extremely rapid evaluations of huge quantities of molecules that may counter the infectiveness of the virus.
So let's go now with some further information about the homology modeling of protein complexes. So let's start with step one. How do we get structures if we only know the gene products? It's trivial to go from gene to protein sequence, simple mapping of 3 nucleobases to 1 amino acid, but I don't want to go in depth about that. From literature, of course, one may also know which proteins act together to form a complex, and input amino acid sequences, wait for a few minutes and get a 3D structure. How this works? By taking advantage of known structures of evolutionary related proteins, and there are more and more of this information that we can have. Here you have some information about the Swiss Institute of Bioinformatics that worked right on the homology modeling. You can find information going to our website, escalate4cov.eu, in the Open Science page, and you have all the link probably you are interested in. Now, going to the druggable proteome, over time, more and more proteins were experimentally determined, which we, of course, used for our study. Furthermore, homology modeling can only be used if a suitable related structure is found. Wherever this was not the case, we used models from AlphaFold, which had proven in the past to be the most accurate method in such cases. Molecular dynamics were performed on each protein to explore the flexibility of the protein and choose conformational states suitable for virtual screening. And now we go through the binding site of Druggable Proteome chapter. The viral protein was subject to a binding site and notation step useful for subsequent modeling studies. As a first approach, research was carried out on scientific publications, which highlighted the presence of conserved binding sites among homologous viral proteins and which regions were occupied by orthosteric and or allosteric ligands. In the second approach, we tested an automated binding site analysis using the binding site found in the literature. And we combined a classical procedure to find a pocket or a cavity on the protein surface and another one based on a docking procedure that uses a ligand as a probe in order to find the druggable sites. Exploring the protein structures in 3D is really a journey and is a challenge. So now the result of the pocket finding method is proposed, pockets with a docking pose. How do we best look at that? Nanome, one of the partners of the Escalate for COVID, is a member of the league that I mentioned before, so supported the Escalate for COVID consortium with some breakthrough technologies of augmented reality and virtual reality. That is basically the next step for the research in the drug design. As you probably know, the augmented reality and the virtual reality allows for intuitive interaction with more complex data and is the next major computing interface. In the past, of course, we worked with 3D visualization, but this lacked the ability to easily interact and modify the structures. And this is the traditional 2D methods. This is a typical representation of the molecular structures that contains, by the way, risk of information loss in structural analysis. This is an image of the virtual reality analysis of binding pockets that we worked at Escalate for COVID. And all this information gave us the chance also to build with the molecules with the functional groups, doing auto energy minimization, the visualization of clashes, and also to understand the hydrogen bonds. And last but not least, the upside that we receive is the virtual co-working space. So basically, researchers from all around the world are working with us and worked with us across sites for engaging presentation and, of course, also design meetings. Now, please watch this video that we prepared with some partners of ours, in particular Gerardo Tauriello from Swiss Institute of Bioinformatics and Edgardo Leila from Nanome that will show to you the works that we did with virtual reality. Wait, sorry.
Here we are now within the VR environment of Nanome, looking at this protein in front of us. It's a protein complex. We see here the red part is a protein called NSP10, and the green and blue parts are two domains of the NSP14 protein. So this is a heteromeric complex of SARS-CoV-2, which takes care of, it has two functions. It has the green domain, which is the exoribonuclease, which takes care of proofreading of the RNA during the RNA replication. And we have the blue domain here, which takes care of, it's a methyltransferase responsible for our mRNA capping. So we have here an interesting sketch of the of how these proteins work together within the larger RNA replication complex. So we have on one hand NSP10 and NSP14 that we have in front of us, and we have next to it NSP16 complex with NSP10, which is another methyltransferase. We have the main RNA replication machinery, which is NSP12, which works in complex with two other proteins not shown on the sketch, which are NSP7 and NSP8. And we have the helicase NSP13, which takes care of positioning the RNA according to the needs of the RNA replication machinery. And so what we have in front of us is a homology model. This was generated with a service called Swiss model, fully automatically, given the protein sequences of NSP10 and NSP14, and it identified as a template structure from SARS-CoV-1, which was experimentally determined by Ma and colleagues, which is also where this sketch here is coming from. And so what we use this for, apart from doing virtual screening afterwards, what I want to show you here at least, is that we use this structure for an experiment to try to identify binding site pockets in this structure. So we did this in two steps. On one hand, we had a literature study where we found all the possible ligands that could bind the various proteins of SARS-CoV-2. And for this one here, we found, for instance, that in the methyltransferase domain here, we have two known ligands. There's a guanosine adenosine triphosphate, which we'll display now. What we see is where we predict this ligand to be bound based on this algorithm. So the algorithm that we used combines two different tools. One called fPockets that finds pockets in general on a given protein structure, and the other is a docking engine. We used one called Plans, which provides with a docking score. It tries to position these ligands that we see here within the pocket, and it provides an energy score. And what we found on this protein and also on many others is that if we combine these two scores and put them in a consensus score, we're able to rank the pockets in a way that it finds the correct one very reliably. And this is the one that we indeed see here. And I'm going to show two other examples. We have one example here, which is basically we have a pocket, which the pocket finding algorithm considered to be a very good pocket, and the docking algorithm trying to position it here really could you could see it rather well when looking at it closely. But it's too tight for this ligand that is trying to put in. As a result, the docking score has a very poor energy, and the rank of the combined score is very poor. And we have the other case here, where the docking algorithm thinks that this fits quite nicely. It's happy about the energetic properties of this position, but the pocket finding algorithm gave it a fairly poor score. And so in this case, the combined score is really able to say, hey, this is not the right position, because the right position, as we know, should be on this side here.
So about the molecular dynamics analysis. So the molecular dynamics is not just useful to provide an input for the next steps, but can provide insights directly too. The same was also seen in other trajectories and when looking at the volume of the pockets. Now I go to a next slide because I know that we do not have much time, and I have some also final remarks that I want to say to you. And I go directly to the virtual screening protocol. So in detail, this is how the parts of the virtual screening link together, starting from the sequences which are continuously monitored if new proteins are discovered or interesting mutations show up. Then we have the structures and homology models with the binding site annotation and the molecular docking mentioned earlier. Finally, the chosen frames from MD are used for virtual screening with a set of docking and genes and possibly metadynamics to explore mechanism of action of PLI. 276 compounds tested in cell-based screen and 4% active, so about 11. Using protocol of 3CL Pro, the screening was extended to all 12 druggable targets of SARS-CoV-2. You see the green more active, red less active. Additionally, to phenotypic screen was performed, including the drugs proposed as active in silico, intersecting the two results. 22 drugs were found, which were then further reduced to raloxifene plus more or less 8 candidates. What next? This is extremely important. So we are now with an ongoing clinical trial for raloxifene at Istituto Malattie Infettive Lazzaro Spallanzani in Rome and Humanitas Hospital in Milan, Italy. We are monitoring SARS-CoV-2 mutations and preparing the tools for other pathogens. But I want to spend a few minutes about the MEDIATE initiative. This is the press release from the European Commission of last October 27 about the green light to the clinical trial in Italy. MEDIATE is an initiative in the framework of Escalate for CoV. Is basically a crowdsourced project virtual screening collection from the pharma computational chemistry and AI machine learning communities. So it was requested by the European Commission to support the European research of the research groups not already involved in H2020 project. And this is the workflow. So Escalate for CoV will make available in this initiative chemical libraries, as you can see, and 3D structures of the SARS-CoV-2, 3D structures of the SARS-CoV-2 non-structural proteins, and many other information useful for the scientist. 10 million CPU time to run the simulation on the Cineca infrastructure, and the web portal to collect the community results. The deployment of this initiative is that will generate a global model for the prioritization of the compounds. We'll use the generated knowledge to acquire up to 10,000 novel molecules, and will experimentally test the compound in phenotypic and biochemical screenings. And the exploitation, so all the groups can publish their results. This is, I think, some very important for the research group. Simulation plus experimental data will be available on a dedicated issue on the International Journal of Molecular Sciences. So we are raising now money for this additional initiative to Escalate for CoV. So if someone among the community of entrepreneurs, philanthropists, and government have an interest to know more, please email me or ask about Escalate for CoV. We are putting together also a scientific advisory board. The Nobel Prize Arye Warshel, that is basically the father of molecular docking with the Nobel in 2013 in chemistry, so is already part of this board. So the board will work on quality assessment of the project, support the Escalate for CoV team in the design of the global model for compound selection, will evaluate the grant proposal for the CPU, GPU time request, and award the groups that performed the best individual prediction with a grant of 1 million CPU hours. So this is a slide with all about the end of the presentation. You can have information about Kelonia Applied Science. That is, by the way, a window also to enter to Escalate for CoV as a leader of the work package on communication and dissemination. But you can go also on escalateforcov.eu where you find all the information. And please join the conversation through our social channels. So let me just stop for a moment the sharing of the screen. I don't know if now you can see me. I know from the technical people that some problem appeared because so the window with my face didn't appear during the presentation. I'm sorry about that. But anyway, so I hope that all was clear. As project innovation manager at Escalate for CoV, I remain at your disposal. What I can say more, so just stay well, stay safe, and enjoy the rest of this great event that is World Innovation Summit for Health. Thank you very much.